Welcome viewers. So we are studying citric acid cycle, and in citric acid cycle so far we have covered step 1 or the detailed mechanism of citrate synthase enzyme which gives rise to the formation of citrate. So we will start from citrate moiety. Okay. So the step 2, which is known as dehydration and further hydration, is catalyzed by enzyme acotinase. Acotinase is an iron sulfur protein but does not contain heme iron. Correct? The dehydration followed by hydration give rise to ultimately isomerization of citrate to a moiety which is produced termed as isocitrate. And as per our experience of isomerization reactions, we got to know that it is not the simple ones as we have discussed in glycolysis. Okay. So why it is important to convert citrate into isocitrate or to isomerize citrate molecule into a second molecule? So the answer lies in the fact that the tertiary hydroxyl group, this is the hydroxyl group, and this hydroxyl group is tertiary here because the carbon is attached to the primary, secondary and tertiary, correct? This tertiary hydroxyl group is not properly located in the citrate molecule, which that's why it does not support the further oxidative decarboxylation which is going to happen in the next step. And in organic chemistry, it is a well-known fact that tertiary alcohols cannot be oxidized while secondary alcohols can be readily oxidized to ketone. Okay. So that's why it is very important to remove hydroxyl group from here and place at the second position. Okay. So the detailed mechanism of this reaction is written below on the bottom of the slide, in which formation of isocitrate from citrate takes place via formation of cis acotinate or a reaction intermediate.
So first, dehydration, the water molecule releases by taking hydroxyl group from the carbon number 3 and hydrogen from the carbon number 2, leaving a double bond between carbon number 3 and carbon number 2, as you can see here, which give rise to the formation of, and this intermediate is termed as cis acotinate, correct? Now further another water molecule comes, and here the hydroxyl group binds at the carbon number 2 while the hydrogen atom to the carbon number 3. Means vice versa happens. The OH from the 3 carbon transfers to the carbon number 2 and simultaneously hydrogen from the carbon 2 transfers to the carbon 3, correct? So these products are the isomers of each other. So why it is important to dehydrate first and to hydrate again? Because to change the position of the hydroxyl group, correct? So by placing the hydroxyl group at the carbon number 2, it is very easy for isocitrate moiety to undergo decarboxylation in the next step. That's why it is important for citrate molecule to undergo isomerization. Okay. Now coming to the step 3, which is known as oxidative decarboxylation, is step 1 of citric acid cycle. Yes. So far we have 6 carbon moiety in our hand. Citrate is a 6 carbon molecule and obviously its isomer is obviously a 6 carbon moiety, and at the end of this step we will have a 5 carbon molecule in our hand. Okay. So this step is catalyzed by isocitrate dehydrogenase, and this is the first of the 4 oxidation reduction reactions of citric acid cycle. This is very important step as there are many first related to this step, like it is the slowest and hence the rate limiting step of citric acid cycle. When we were studying enzymes, I told you that the slowest steps are the rate limiting ones. Okay. Second, it produces the first NADH of the citric acid cycle, and obviously it is the first decarboxylation reaction of the cycle too. Correct? Now let's focus on the reaction. 6-carbon isocitrate is acted upon by NAD+, which give rise to the formation of reduced form of NAD+, which is NADH plus H+, and again an intermediate formed which is known as oxalosuccinate, and this intermediate is also a 6-carbon. Obviously there is no decarboxylation in the step 1. Okay. Now in the step 2 means after formation of the intermediate, decarboxylation reaction happens, and the blue COO minus removed in the form of carbon dioxide, leaving a moiety, a 5-carbon molecule which is known as alpha ketoglutarate. Correct? Okay. One important point which is related to the intermediate oxalosuccinate here is that it is both an alpha keto acid and a beta keto acid. How? See, focus on this carbon, the COO group. Okay, focus on this group. So any carbon attached to the functional group is known as alpha. So if this is a functional group, the COO minus, this carbon is termed as alpha. But there is another COO minus in the neighborhood of this carbon. Okay, the blue one. So as per this carbon, this functional group is called beta, because directly attached carbon is alpha and second carbon is beta. So as per this COO minus, this is alpha. So as per this COO minus, this is beta. Okay. So it is a very important intermediate because it is both an alpha keto acid, keto because it has COO group, and a beta keto acid respect to this blue COO minus. Okay. So but because of the beta ketoic property, it allows the loss of carbon dioxide in the enzymatic reaction in the conversion to the 5-carbon molecule. Are you getting my point? What I was trying to say is that isocitrate loses COO minus in the form of CO2 to generate alpha ketoglutarate, correct? But this does not occur immediately. As you can see, when isocitrate has aldehyde group, the OH group, it doesn't occur immediately. Rather, it is required to convert the second carbon into a keto group first, and because of the beta ketoic property of the oxalosuccinate, although it has alpha ketoic property also, because of the beta ketoic property of oxalosuccinate, it is easy for this moiety to remove the second COO minus group in the form of CO2 to undergo decarboxylation easily to convert 6-carbon isocitrate into a 5-carbon alpha ketoglutarate, correct? So remember every tidbits which I am explaining in every step. Okay. So ultimately we have 5-carbon alpha ketoglutarate as a product of this reaction, and in the next step this alpha ketoglutarate molecule further undergoes decarboxylation, and which is termed as oxidative decarboxylation step 2. Why we are taking oxidative term again and again? Because this citric acid cycle reactions are taking place in the mitochondrial matrix, and matrix is aerobic. Okay. So this step is catalyzed by alpha ketoglutarate dehydrogenase complex. Have you ever heard about a similar complex before? Yes, we have heard about pyruvate dehydrogenase complex, and we have gone through the detailed mechanism of PDH complex converting pyruvate to acetyl coenzyme A. So when you see the two reactions parallelly, you might get an idea that these reactions are very similar, as both in the structure of the substrate as well as the cofactors and the releasing molecule. Okay, it was pyruvate here in PDH, it is alpha ketoglutarate here in the alpha ketoglutarate dehydrogenase reactions. Okay, NAD plus used as a cofactor along with coenzyme A SH, also these two are cofactor here, and the resulting molecule is acetyl coenzyme A here in the PDH, and it is succinyl coenzyme A with a released CO2, correct. So these, because these two reactions are very, very similar. Why? Because the enzymes catalyzing these two reactions are very, very similar. Yes, alpha ketoglutarate dehydrogenase is very, very similar to the pyruvate dehydrogenase complex. The only difference is that it doesn't have the two regulatory enzymes which PDH has. Okay. So if you remember the PDH complex components, you might remember that it has E1, it has E2 and it has E3, the three enzyme, the three catalytic subunits you can say. Okay. So when it comes to alpha ketoglutarate dehydrogenase complex, the function of E1 is to pick up the 5 carbon alpha ketoglutarate moiety and to remove carbon dioxide and converting it into a 4 carbon fragment and holding its electrons. See, whenever a bond is break, electrons are released. Okay, so these electrons are holded up by the E1 complex, correct. Now the E2 complex, what does it do? It picks up the 4 carbon fragment from the E1. You remember it has a lipoamide arm which acts like a crane. I explained in the PDH video that it picks 4 carbon fragment because of its lipoamide arm and electrons also, and deposit onto coenzyme A, creating thio-S coenzyme A. Means what is this? Thioester linkage. Okay. Now the third unit comes into place, into action, and it finally picks up electrons and also NAD plus to form the second NADH of the citric acid cycle, correct. Similar. So the decarboxylation reaction of an alpha keto acid, the alpha ketoglutarate is an alpha keto acid. The carbon atom directly attached to the functional group is known as alpha. So it's an alpha keto acid which give rise to the formation of a high transfer potential thioester linkage with coenzyme A, forming coenzyme A acid. Sorry, forming succinyl coenzyme A, correct. So it is not necessary to explain here that this reaction will move towards in the forward direction only, because it requires or it leads to the formation of a high transfer potential thioester linkage. Okay, we have explained many times. So I am ending this lecture over here so that you will carefully watch these videos and revise every step which I have explained about the nature of every step, about the nature of every enzyme, and about the nature of every intermediate form. Okay, happy learning.